Hello everyone, my name is Corinne Gillion and I'm a senior biology major here at Presbyterian College. Thank you so much for joining me for my Honors Day presentation, and today I will be presenting my research entitled Embryogenesis in the Live Bearing Fish Phocelia latipina, a correlative microscopy study. So this is an outline of my presentation today. I'll begin by stating my goals and objectives, including two experiments that I was able to conduct and how those experiments relate back to the overall goal of this research. And for the first experiment, I was able to classify normal stages of development of mollies. I'll start by going over introduction, and then this will lead me to materials and methods, including molly breeding and husbandry, scanning electron microscopy, photomicrography, and histology. I'll state my results and give a brief discussion. And this will lead me to my second experiment, which deals with the absorption pattern of iron for mollies. I'll go over a brief introduction and materials and methods, including molly separation and iron-fortified diet imaging. This will lead me to my results and discussion. And then finally, I'll state some future research and give my acknowledgments and references, and then open the floor to any questions. So the overall goal of my research was to see if mollies are model organisms for maternal-fetal transport. And so the first step we needed to take to be able to do this was to classify the normal stages of development for mollies. And to do this, we used correlative microscopy techniques. And in the next slide, I'll go over some of these techniques that we used. And then the second step, after we classified the normal stages of development for these mollies, was to look at the absorption pattern of various nutrients that are passed from mother to fetus. And so we looked at iron as our first nutrient, and to do this we were able to look at histology slides of the gut and use differential staining to see how iron is absorbed in the molly gut. So this leads me to my first experiment for my research, in which we classified the normal stages of embryonic development for mollies. So now for a little background information on mollies. Phocelia latipina, also known as mollies, are a viviparous species of fish. This means that they give live birth, and this is in comparison to many other species of fish that are oviparous, meaning they give birth to eggs, and these eggs mature in the external environment. So because mollies are viviparous, the pregnant females have a maternal brood pouch, and this brood pouch is an enclosed environment that allows many different nutrients to be delivered from the mother to the embryos. And because these mollies have a maternal brood pouch, we wanted to test and see, is this brood pouch analogous to the human uterus and other mammalian brood pouches. So one of the questions we had to ask ourselves is, why is this important? And so when we started looking in the literature, many other species of fish, their embryonic development has been well documented for several years, including the Medaka, and these scientists have been able to collect images which are kind of comparable to the Carnegie stages of development of human embryos and fetuses. So we wanted to be able to do that with mollies because no series of images like this has been collected for any oviparous fish that we could find in the literature. So to begin this experiment, we prepared two tanks with brackish water. Mollies are tropical fish, so they live in salt water, so we simulated their aquatic environment. After preparing the tanks, we purchased 24 mollies from Petco and then separated them into the tanks. We wrapped each tank in aluminum foil and used poster board to prevent any light from entering the tank. And then we used photomanipulation by having lights set on timers in the tanks. And then so the lights had 8 hours of darkness, 8 hours of light. And so this sped up the breeding process so that we would be able to capture as many stages of molly development as possible in the amount of time we were given. Once the mollies were left to bred for about a week, we came back and were able to see any pregnant female mollies. And so once we identified those mollies, from a kind of puffy maternal brood pouch. We gave the mollies a cesarean section. To do this, we anesthetized the female and with MSDS, and then we extracted the pups, and then we put the female in a recovery tank for a few hours, and then once we knew she was good and swimming well, we put her back in a regular tank to heal. And with the pups, we divided them into 3 groups. Usually the mother was able to produce around 6 pups, so we had 2 pups in each group, and we did a different correlated microscopy technique on each set of pups. So the first set of pups underwent scanning electron micrography, and the first step of this process is dehydration, and this just removes any water from the embryo and allows critical point drying to happen a lot quicker. And this is the setup for the critical point dryer. And then once these embryos are dried, they're sputter coated with gold so that we can see all of their external features, and this gold reacts to the with the scanning electron microscope, and then you're able to see detailed images of the embryos in their external anatomy. The second set of pups underwent photomicrography, and the first step in this process is dehydration with ethyl alcohol. And then once the embryos were dried, they were placed under a microscope, and color images of the external anatomy of each embryo was documented. So the last set of pups underwent histological preparation, and this process was definitely the most time-consuming and took the most amount of trial and error. We began by dehydrating these pups, and then this led to the embedding process. So we experimented with xylene and paraffin embedding versus JB4 embedding. Xylene and paraffin embedding worked really well for small embryos, but as the embryos got bigger in size, a lot of times the cuts on the microtome with the xylene paraffin blocks would fall out, or they just wouldn't hold together, and the cuts would be jagged, and so they wouldn't be able to really see anything once we did histological staining. But when we used JB4 embedding, we found that the embedding solution was much more incorporated into the embryos and was able to have a really distinct cut, which allowed it to be easier to stain these segments of the molly, and then we were able to clearly identify internal structures based or with the differential staining techniques that we used. So this leads me to the results of my experiment. We were able to classify 7 stages of development for mollies in this experiment. And the first stage was the unfertilized egg. The process that is occurring in this stage is voletinogenesis, which occurs when yolk granules are deposited, and this creates a barrier for the embryo and eventually will be food for the embryo once it is fertilized. The second stage of development is cleavage, and in this stage the yolk is being pushed to the periphery and the blastoderm is forming. So the blastoderm will become the embryo, and so it is protected by that layer of yolk, and this happens about 30 minutes after fertilization. And this leads to the third stage of development. In this stage, organogenesis is occurring, which is the creation of the organs and organ systems. And one process that occurs in this stage is neurulation. So, as you can see in this image, the optic cup is forming, which will eventually become the eye, the choroid, which is the inner part of the optic cup, and the pigmented part of the eye is beginning to form, as well as the retina. And then in this histological image, you can see where the optic nerve is starting to grow into the eye, and then you have your optic vesicle. And so for this optic cup to form, you have to have contact between the optic vesicle and the epidermis, and this leads to an invagination, which creates the optic cup. And when the optic cup becomes more differentiated, it will have a lens placode in the middle, which will eventually become a functional lens. Another process in this organogenesis stage is cardiogenesis, which is the creation of the heart. As you can see here, there's an atrium and ventricle. Angiogenesis is also occurring, which is the creation of blood vessels. As you can see here, you have two primary blood vessels coming off the heart. And then hematopoiesis is also occurring, which is just the creation of red blood cells, as you can see in the red coloration in the embryos. And then the chorion is also present, which is a protective layer around the embryos, and this just prevents anything from entering the embryos that should not be there. So the fourth stage of development is the hatching stage, and this is when the embryos enter the maternal brood pouch. And in this stage, you have pigmentation of the choroid and retina. And what this does is helps protect the eye and underlying brain from injury from UV light. Also, you have early chromatophores forming, and chromatophores are neural crest cells that migrate to various places on the body of the embryo and also protect it from UV light damage. You have a decreased yolk volume as seen here, and this is compared to the massive amount of yolk volume in the egg, and this is decreased as the embryo begins to mature and eventually will not have a yolk volume and need to make its own or be able to gather its own food. You also see here that a fin bud is forming, and this will eventually become a functional fin. As you see here in this histological picture, you have the lens placode fully formed, the choroid, which is pigmented, and also you have the retina. So the fifth stage of development for mollies occurs when many internal structures are beginning to form. Here you see a functional gut tube. You also have the heart and two primary blood vessels, the efferent arterioles and afferent arterioles. So this is the primary blood vessels, but then also in this stage you're seeing the amplification of blood vessels into secondary and tertiary vessels, as you see here and over here. And this just supplies blood to various regions of the embryo. You also have the liver being formed and the notochord. As far as external structures, you see gill rakers, which will eventually become functional gills, and then osteocyst. An osteocyst is an organ that is analogous to the inner ear of a human, so it senses vibrations and movement and is able to detect pressure as well. And this really helps the fish, once it's expelled from the maternal brood pouch, to be able to sense its environment. And this leads to the sixth stage of development, which is the post-hatching stage. And in this stage, the embryo has been in the brood pouch for a while and is beginning to really mature and eventually be ready for partition from the maternal brood pouch. In this stage, you have your differentiation of your brain, including the telencephalon, diencephalon, and met and myelencephalon. And so this is, in layman's terms, it's the forebrain, midbrain, and hindbrain. And so you're really differentiating parts of the brain for various functioning. And so the first part of the brain would be more thinking, and then the hindbrain would be like survival skills such as breathing, swimming, that kind of thing. You also have lepidotrachea, which will eventually become fins, and then you have lateral myotome being formed, which this is just specific banding of the muscles so that they are able to move faster. You also have a lateral line that's beginning to form, and this is an organ that allows the fish to kind of tell where they are in the water, so it allows for pressure sense and kind of depth of water that they're in. So this leads to the final stage of development, which is the pre-partition stage. At this stage, the fish is at full maturation and is ready to be expelled from the mother's brood pouch. And here you see a fully functional eye with the optic cup and lens placode. You also see that the fish has a decreased yolk reserve in comparison to the previous embryos. And this is because the fish is about to be expelled and go into the environment, and so it should be able to get nutrients on its own. You also have these organs here, which are neuromasts, and these are part of the lateral line organ system. These are mechanoreceptors that allow the fish to sense vibrations and movement so they're able to detect predator and prey. This is a scanning electron micrography picture of it, and as you can see, it has many of the same, looks the same as a hair cell in the human ear. It serves the same purpose, but detecting vibrations and pressure and all that kind of thing. So from this experiment, we were able to classify 7 stages of development for mollies. And we were able to do this using the internal and external structures that we were able to photograph using correlated microscopy techniques. So potentially, if we had a random molly that we didn't know how long it had been maturing or anything like that, we would be able to tell about what stage it's in based on its internal and external structures. And in the future, we can use this normal stages of development as a reference guide for when we test various agents, pharmacological agents, or carcinogens on these mollies and see how these agents affect development of mollies. And this leads me to my second experiment, in which I looked at the absorption pattern of mollies. And so going back to our goal was just to see if mollies are a model organ system for maternal fetal transport. We need to see how are nutrients absorbed in the molly from mother to fetus, and is this analogous to humans and mammals that are we trying to compare it to. So now for a little background information on absorption patterns in various animals. So mammals have regional absorption of nutrients. As you see in this figure, you have the small intestine, which absorbs many nutrients such as amino acids and carbohydrates, and then you have the large intestine, which is mainly involved in salt and water absorption. This is in comparison to fish, many oviparous fish that have one intestine, and so this intestine absorbs all nutrients, and there isn't a regional absorption but a uniform absorption of nutrients. So to be able to validate our hypothesis that mollies are a model organism for maternal fetal transport, we would need to see what type of absorption pattern they have, whether it's closer to mammals or whether it's closer to more oviparous fish. The materials and methods of this second experiment are very similar to the first experiment. So we divided the mollies up into 2 tanks and we left them to breed for a week. We used the same photo manipulation techniques that we used in the first experiment, and the water quality was the same. One difference between the second experiment and the first experiment was the diet of the fish. So in the first tank, the control tank, we fed the fish just a normal diet of tetramin fish food, and then the second tank had fish that were our experimental group that received an iron-fortified diet. A previous student had made this iron-fortified food mix, so I used that mix for the experimental tank. Once the pups were born, we extracted them from the water, and then they underwent histological preparation. And once the histology slides were made, they were viewed under a light microscope and were able to determine the types of absorption patterns that these mollies had. So this leads me to the results of my second experiment. As you can see, the mollies that were fed the normal diet, or the control group, have no iron found in their foregut, and they have no iron found in their hindgut. When looking at the results from the iron-fortified diet group, you can see that these fish had iron in their foregut, as seen by these small black dots in the foregut. But when looking at the hindgut, there's no iron in this region. And so this leads me to my discussion of my second experiment. So we were able to determine that mollies have regional absorption of iron in their gut, which is similar to mammals. So we came to this conclusion by looking at the normal group, the control group, and they did not have iron in either their hindgut or foregut. In comparison to the other group of mollies, the experimental mollies that were fed iron, and they had iron in their foregut but not their hindgut. Therefore, we concluded that these fish have regional absorption of iron. So in the future, we need to look at the absorption pattern of other nutrients such as carbohydrates, proteins, to see if this regional absorption of nutrients continues for the other nutrients. And so this would further validate our hypothesis that mollies have regional absorption of nutrients and therefore can be used as a model organism for maternal fetal transport. So now that we know that mollies can be potentially used as model organisms for maternal fetal transport, and we know their normal stages of development, we can test the effects of various pharmacological agents on the development of mollies in the maternal brood pouch and maternal fetal transport itself. So one of the pharmacological agents that we are considering is triclosan. So triclosan is known for its antimicrobial properties. It's found in a variety of different products such as hand soaps, creams, shaving cream, hand sanitizer, toothpaste. So it's found in many of the products that we consume daily. Scientists have found and have done previous research that found that triclosan is an endocrine disruptor. So this means that the receptors that are supposed to react to natural hormones are either being blocked or changed so they cannot react to this natural hormone. And as we know, hormones are very important during pregnancy as they signal various structures to form and when to develop, and it's important in the timing of development. So our future research could focus around various levels of triclosan and seeing if it has effect on the development of mollies. So now I'd like to acknowledge some very important people that have made this research possible. First, I'd like to thank Dr. Wetzel, my advisor. He's been crucial throughout this whole process, answering questions, teaching me things, getting me on the right path when I go astray. So he's been very influential, so I really appreciate him. Also, Tiffany Bogan, she kind of started this research and left a lot of great notes and things that helped me get the process started a lot quicker. She also made the iron fortified diet food mix that I use, so thank you, Tiffany. Also, SCMBRE, they sponsored my first experiment, which was done over the summer, and specifically thank you to the whole Presbyterian College Biology Department for their help through sophomore seminar, junior seminar, getting me prepared for this presentation. Thank you to everyone. And then finally to my family and friends who have stuck with me through everything and encouraged me and have really made this process great for me. So thank you to everyone. These are my references that I used throughout my research process, and many of the papers that I cited are listed here. So this concludes my Honors Day presentation. Thank you so much for sticking around and listening to me. If you have any questions, feel free to email me at cegillion@presby.edu. I would be happy to answer any questions or to Google Meet and answer any questions. So thank you so much again for watching and have a great day.